This topic will be covering protein tertiary and quaternary structure. So in a previous topic, we discussed the primary and secondary structural aspects in terms of protein structure. And so we're going to build upon that in higher order structures. The learning objectives of this topic are shown below. So primary structure of a protein is its amino acid sequence. The secondary structure of proteins are primary alpha helices and beta sheets, and those we described in a previous topic. What we're going to cover now are the secondary protein structural elements as they fold together to form a 3D protein structure called the tertiary structure, and the role of domains in this structure. So we'll be defining what a domain is. And then the latter part of the topic will be looking at higher order structures, quaternary structures, which are various associations of two or more folded proteins that have a tertiary structure into a higher order complex. And some protein complexes can get quite large. And so you'll be seeing examples of these types of proteins and their structural features and how it relates to regulation of the proteins in metabolic pathways as we move through the course. But right now we're going to look at those structural characteristics of tertiary and quaternary structure. Tertiary structures are basically the three-dimensional structure of a polypeptide chain that has folded into a stable structure. And so we can envision the peptide chain that's being formed, and the backbone is white, and that the side chains can be classified as either being polar or nonpolar. And what happens to drive protein folding, which will be discussed in a different topic, is that these, and we can measure this experimentally and look at protein stability, is that the nonpolar side chains, the hydrophobic side chains, will want to interact through hydrophobic interactions, and these will be buried on the inside of the protein, and then the hydrophilic side chains will be on the outside. And so as structural elements start to form on the secondary structural side with alpha helices and beta sheets, they'll also come together in terms of their side chain interactions to have this hydrophobic core and hydrophilic surface. So recall the hydrophobic interaction just to acquaint yourself with the thermodynamic considerations as to why a protein folds with the hydrophobic core on the inside and the hydrophilic surface on the outside. So if you have a molecule that has a hydrophobic component as shown with a lipid, as we did this example in the thermodynamics section, is that water molecules in the aqueous solution have to hydrogen bond in such a way as to create a cavity in which to accommodate this hydrophobic region of this molecule. So this water molecule that creates this cavity is very ordered. And if we have several of these in solution, and as we increase the concentration of the lipid, the hydrophobic interactions will tend to interact with one another, and water will be displaced. The hydrophobic regions will interact through van der Waals interactions, and a new cavity would be created to accommodate this ensemble of lipid molecules. And if we look at the overall amount of ordered water surrounding an ensemble of lipid molecules versus individual lipid molecules themselves, there's much more ordered water here than here. And in that case, the entropy of the system is greater. There's more disorder in this system because there's less ordered water molecules than here. In addition, we have van der Waals interactions that are favorable, and that helps contribute to promoting the interaction. Although we had to break a lot of hydrogen bonds here, the amount of disorder we get in this process drives the lipid molecules to interact with one another. And this is the hydrophobic interaction. So the same thing applies to proteins, in which the hydrophobic side chains of the amino acids will have the same function happen. Where if you think of the secondary structures that we talked about in terms of alpha helices, in which the side chains point out from the helical axis, and the beta strands, where you have the sheets formed and the residues of the side chains are either on one side of the sheet or the other, they can be hydrophilic or hydrophilic, and so they will have to be accommodated if they're hydrophobic with water to surround them and be ordered. And so if you were to drive these hydrophobic residues together with different secondary structural elements in the process of forming, you will get these hydrophobic core sequestered from the solvent, driving protein folding. So we'll talk more about this again in terms of measuring this experimentally and what kind of stabilities you get through this type of protein folding mechanism. But let's just say at this point it promotes the stability of a folded protein, but it's not too stable that the protein can't be unfolded and degraded because it could be used for their amino acid content if we need it for energy source, and if a protein is no longer functional, we need to get rid of it if it's a problem. So we want it to be stable but not too stable. So we'll talk more about that in the folding section. I want you to understand the hydrophobic interaction again is being realized here when we're talking about proteins going into these tertiary structural three-dimensional entities. This is an older slide that I had when I first started teaching here, but the numbers may probably be a little different. But if you look at the protein data bank and look at the accessibility of residues of amino acids in terms of solvent accessibility or buried on the core, if we say accessible is at least 5% or more of their side chains are accessible to water in terms of its surface area. 27% of these are charged, 34% are polar, and surprisingly, there's a lot of nonpolar residues that are present on the molecule. Overall, it's 60% polar, but it's 40% nonpolar. So it's kind of like, that's a little surprising. Let's see what it's like on the inside. Do the same analysis on the inside, and you can find that it's 78% nonpolar. There's some polar components on the inside too, because again, it's not 100%. Things get trapped as some hydrophobic regions start to fold. It'll pull some polar ones along for the ride because they happen to be part of a helical region, for example, that's kind of forming as part of its side chains into the core. There may be a residue there that's polar or charged, and so you could have some of these things kind of part of an internal part of a protein, but not a lot. 78 nonpolar, okay, kind of makes sense, but the surprising, I guess, value would be the nonpolar. And nonpolar interactions are important for driving protein-protein interactions, not necessarily charge-charge interactions. And so the same hydrophobic effect that goes into folding the protein to have more of a nonpolar or hydrophobic core also comes into play when we want to have two protein molecules go together to form some quaternary structure. There's got to be some interaction between those two macromolecules, and so hydrophobic interactions can play an important part of that association. And so there's quite a rather high percentage than you would think of nonpolar residues on surfaces that will help drive that. Again, these nonpolar residues aren't just randomly distributed throughout the surface, but they're usually regionally confined to provide a hydrophobic face that would allow the interactions to occur between proteins. So Again, there's a lot going on here in terms of protein structure and function. But the overall concept about the hydrophobic core versus the hydrophilic surface is pretty much preserved, but not as high as a percentage of hydrophilicity as you would think that would be on the surface of a protein. There are different ways to depict protein structures, and so one of the common ways that's very informative as regard to secondary structural elements is the cartoon model, or what's also known as the ribbon diagram. And that's where you see these ribbons that will be used to define an alpha helix or your beta sheets. And then interconnecting loops and turns are shown as just these linear renderings. So in this case, you can see how this sheet is a 1, 2, 3, 4, 5-stranded sheet, and it's in an anti-parallel fashion. So the arrows help you know where the end terminus and C terminus end of the sequence are, and you can easily identify if a beta sheet is anti-parallel or parallel. Another way to look at structure is to look at the stick model, and if we zoom in on a particular region, you can see the backbone. In this case, the carbons are in green. We're not showing any hydrogens here, so it's kind of like a chicken wire diagram in that sense. The carbons are green, the nitrogens are blue, and the oxygens are red. So if you look at this particular residue here, you have a CH2, a CH2, a C, an oxygen, and a nitrogen. So do you recognize what amino acid residue that is? It has a CH2, CH2, C double bond O, NH2 side chain. It's glutamine. So these are the things I want you to think about, learn the structures, is to recognize key features of proteins. It makes it easier to follow what's going on, especially when we talk about mechanism. And we'll do that for one example in this course in a different topic. But understanding knowing the structures of the amino acids helps you figure out what's actually happening in the given region of the protein as well. You can see hydrogen bonding interactions showed as this dashed line, and you can get distance information here. You can see, yep, it's within hydrogen bonding distance around 3, so you're good to go. Nice hydrogen bond interaction between this nitrogen amide. It's implied there's a hydrogen here, hydrogen bonding to this carbonyl carbon part of the backbone of the protein chain. We also can look at the surface models, looking at solvent accessibility, what's accessible to water, and so that will be more of a contoured surface, showing you the surface contacts between the protein and what water can access. And so keeping the same color scheme as we had in the previous slide, you can see there is a lot of carbon exposed, as a lot of the hydrophobic component, that carbon hydrogen accessibility there on the surface does convey it somewhat of hydrophobic regions that could be present. You also have a lot of oxygens and nitrogens, so you're going to have a lot of charged surfaces there depending on their side chains. They could be deprotonated and charged, or protonated and charged, and so we can look at those in a different type of rendering called this electrostatic surface potential here, where blue is positively charged, red is negatively charged, and whites are neutral. So you can see there are some neutral regions here. On this particular protein, it seems like half of it's positively charged, half's negatively charged. I don't know what it looks like on the other side, but it could be possibly very similar in terms of the distribution there. If you wanted to just emphasize the surface aspects, where are the irregularities, you would just make it one color. I like the electrostatic potential. That shows you the interactions that are possible in terms of different monomers. How would they interact? You can imagine if you're going to interact with this one, there's not a lot of hydrophobic surface, so it would require the positive end to interact with the negative, the negative features to interact with the positive features of another protein. So these are the surface, solvent accessibility surfaces that we're looking at here. Another way is to look at a space filling model, and the color coding here was the charged residues were in blue, hydrophobic residues in yellow, and white would be other residues that aren't either charged or hydrophobic. They could just be polar. And so you can see here with myoglobin, there is quite a bit of hydrophobic residues that are available to the surface on this face of the protein. You can go through and you can chop the molecule in half, do a cross-sectional look at the interior of the protein, and you can see, as I mentioned earlier a few slides ago, that the interior of a protein is mostly hydrophobic. And indeed, when you do one of these cross-sectional views, you can see that is indeed the case. One other aspect of proteins I want you to remember, and we did talk about before in a different topic, is that the proteins in aqueous solution are surrounded by water molecules, and these water molecules are often tightly associated with the protein through hydrogen bonding interactions, that it creates a hydration shell around the protein. And this is an example shown here for lysozyme. If you crystallize it and do X-ray diffraction to get the electron density map to diffraction map to figure out the electron density of the protein and its structure, water molecules are clearly visible and are shown here. So you can see a lot of the water molecules are tightly associated with the surface of this protein. This is very true for proteins in general. A lot of water molecules surround the protein, help make it soluble. Okay? The interactions that can occur with proteins through the displacement of this water to promote protein-protein interactions would help promote their interactions through a hydrophobic effect as well. So creating more disordered water in the process of proteins associating would help drive the association between those two protein molecules. So don't forget the hydration shell that proteins have. Sometimes these water molecules are so important that they are intervening in the interaction between two protein molecules. So I've seen where you have protein A and B interact, and in the interface of protein A and B, there'll be a series of very ordered water molecules that are hydrogen bonding, hoping to bridge the hydrogen bonding between one protein and the other, mediated through a water molecule, because the space is too far apart for the proteins to do it themselves. But you put a water molecule in to kind of help facilitate that, so there'll be a hydrogen bond, say for example, acceptor on one molecule and hydrogen bond donor on the other, but there's a OH in there from water that helps make that connection, a bridging effect, if you will. So water is very important, and we'll see the role of water important for proteins and their active sites and how they catalyze chemical reactions as well. So don't forget, water is there. Often these structures are shown without it, but if you go back to the actual coordinates that are posted on the protein data bank, and you look at and say, let me see the waters, you'll be surprised of how many ordered water molecules are stationary on the protein surface. So proteins are very diverse in terms of their structures, and we can break them down into different elements, just like we did with secondary structures. There are certain types Well, there's also certain types of protein structural elements, and one is the domain. So you should know what a domain is. It's a compact unit of protein structure that is usually capable of folding into a stable, independent entity in solution. In other words, it's usually a part of a protein that can fold independently. So if you have, say, a protein amino acid sequence of 500 amino acids, one domain may be residues 1 to 200, and another domain may be residues 250 to 500. And so they'll fold independently. So if you were to basically only make the first 220 amino acid residues connected, that will fold as an independent domain. So proteins often have multiple domains, and sometimes they can be quite complex, that they may contain other parts of the protein, not necessarily the contiguous amino acid sequences that are adjacent to one another. So here's an example here showing this leucine binding protein, where part of this domain is formed from a chain that is part of a loop in the other domain. So if this thing loops out, it's actually going to be interacting with another part of the protein to form this domain. So it can get a little more complex too. It doesn't have to be the contiguous segment of the peptide chain. But know what a protein domain is. Here's just an example that we can look at proteins in terms of what domains they contain. So a domain often may have a certain function. So for example, the serine protease domain is a domain in which a serine side chain helps to cleave proteins in this domain. So it's a catalyst. It cleaves proteins. And so you can add that domain as part of a newer protein, like a component. So I want my protein to have a serine protease activity. So you can have a serine protease domain. And then you may want it to have the ability to bind calcium. So you can add a calcium binding domain. And so there's different domains that exist by themselves that can be catalytically functional, but they can be part of a larger protein as part of a domain of its structure. So some proteins have multiple domains. Some proteins itself is the domain. And so we don't really think of it as a domain. We just call it the protein. And so it just depends on what the protein is and how it relates to other structures. So these domains we start seeing the same structures over and over again, and they do the same function, and they get a domain name associated with them. They have some definitive sequences that are going to be the same. So if you did a sequence alignment with different proteins, you would find that they contain very similar amino acid residues. They may differ a little bit, but you can say, aha, that's a serine protease domain. So I know this protein has one of those probably in it, and a calcium binding domain. So the sequence can tell you a lot of information of what domains may be present and what its function may be. But until you actually determine the structure, you can do modeling, but you actually have to have a structure to really know for sure. And so over the years, as more structures were determined, more inferences of domains and reliability based on their assignments based on structure have been developed. So it becomes more computational as opposed to doing actual structural determination. But there's got to be some way to verify those models as well in the experimental setting. So I'm just going to cover a couple common domains or what I'd call motifs, which is kind of a variation of a domain. And sometimes they're called motifs if you see a lot of them that are part of a higher domain. So they're not a single secondary structure, but they're not necessarily large enough to be a domain, but they're like a smaller structural ordered system, and we'll see them a lot, and we call them motifs. There's a lot of motifs. So I'm not going to spend time having you memorize them, but just want to show you a couple examples that you see a lot. And you'll see this throughout the course as well. So if we have two helices, they can come together to form a helix-helix or coiled-coil interaction. And the way that these interact is that one side of the helix contains hydrophobic residues, which interacts with the other side of another helix that contains hydrophobic residues. And so these hydrophobic residues will come together through hydrophobic interactions to form what we call a coiled coil. So you can imagine two helices just coiling around each other. And the other remaining residues that are sticking out from the helix are going to be more polar or hydrophilic, and they're going to be interacting with the surrounding solvent. And so we call such a helix that's half hydrophobic and half hydrophilic amphipathic. So we talked about amphipathic before in terms of small molecules having half hydrophobic and half hydrophilic components. Well, the same thing can happen for a helix. It can be half hydrophobic on one side and half hydrophilic on the other side. In this case, the hydrophobic is here in the interior, the hydrophilic's on the outside. So they have both polar and non-polar character. The leucine zipper is shown here as a motif you see quite a bit, especially when you're looking at transcription factors involved in gene regulation. Okay? And usually they have a series of repeating units to allow these hydrophobic residues to basically reside on one face of the helix. Now, in looking at this coil-coil interaction that was on the previous slide, if we were to look down the helical axis, look from the top, we would see something that looked like this. You have helix 1 over here, helix 2 over here, and then you have the hydrophobic groups, and these would be your side chains sticking out and interacting through the hydrophobic interaction, and then you'd have some hydrophilic residues here, okay? You could also have a situation where you could have, say, a residue here that, say, were a carboxylate side chain like aspartic acid, and then you could have, say, a lysine over here, and you could have a charge-charge interaction as well to help promote the interaction of the helix as well, because the side chains can extend out a little bit. So it can get quite complicated, but I want you to consider the fact that we can look at helical regions based on knowing their helices and their sequences here. We can use the helical wheel to help us determine if the helix is amphipathic or not. All right, so if we look at this rendering here, and we call residue A as the first residue, and we're trying to look down the helical axis here, we know there's 3.6 residues per turn, and we know a circle has 360 degrees, so that means that the next residue should be at 100 degrees from A. So it wouldn't be, this is not B, but where's B? Right over here. And then where's C? Here. D, E, F, G. And if you start back H, it would start back over on A. Because you have 3.6 residues per term, and if you have 7 residues, that kind of brings you back at the beginning, because 3.6 times 2 is around 7. So it kind of takes you kind of around the beginning there. So often these repeats that you see here of leucine, valine, leucine, you have asparagine in here. It's polar, but it's got a couple, has a methylene group in there, so sometimes you can get those in there as well. But these hydrophobic ones are driving the interaction. We'd call this a heptad repeat because it's repeating 7 times. So you see these heptad repeats. So in the sequence, you'd say, I'm seeing this heptad, the sequence of 7 amino acids repeating over and over again. So it tells you something about the helix, if it's amphipathic or not, and if it's able to interact. So if you have hydrophobic faces, it can be able to interact. Another way to look at a helical wheel diagram is shown here. It's kind of like the old spirograph, if you ever did that as a young child and doing the spirograph where you draw, put a pen in a hole in one of these gears, and as you travel along the frame where you have that gear, it will dictate where that pen's going to go, and the offset of the gears will allow it to go into unusual configurations like this. So if you follow this 100 degrees, this would be your first amino acid here, D. Then you have to go over here to see L, then T, then G, then L, and so forth. And so when you look at the residues, you can say, look how hydrophobic this is over here. Look how hydrophilic it is here. It's amphipathic. We draw a line. There we go. I mentioned glycines aren't usually seen in helices, but they can be, and they can be considered hydrophobic because it's just hydrogens there. So if I were to draw a line, it's like, there you go. There's the polar side, and there's the nonpolar side. So I want you to be able to recognize if a helix is amphipathic or not, and using the helical wheel to help you determine that. So there's some problems in the problem set to look at helical wheels and to do that. So it's a very useful tool to look at helix motifs and to understand how they're interacting through these hydrophobic interactions. We can go another step further and put 4 helices together, as seen in cytochrome B562, and we can look at how these helices are arranged in what's called a topology diagram. So it's just showing the secondary structural elements in their connectivity. Very useful for beta sheets. And in terms of this topology diagram, you'll have arrow directions from N to C terminus, and you can see how these helical elements are arranged you can consider that an anti-parallel arrangement, where here you have 2 parallel helices and 2 parallel ones in this direction. So you can see, to keep it in the same N to C direction, you have to have a huge intervening connecting component in order for that to be achieved, and you can see that here with this large interconnecting unit here, and it's kind of shown in this topology diagram. Kind of hard to see in larger structures, but the topology diagram can be very informative as how there is connectivity between the secondary structural elements. Here we have the helices arranged in an anti-parallel fashion, so the turns the transitions between these structural elements don't have to be as large. Also notice that, remember, the helix has a dipole from the N to C terminus, and so these arrows also reflect the dipole direction as well. So these dipoles are lining up in a way that's also favorable for the interaction as well. And you can imagine, if you look down the top of this, you'd see 4 helices bundled together, and then you'd have hydrophobic groups on the inside and hydrophilic groups on the outside. So this is the 4 helix bundle. So these are what we consider alpha proteins. They're all alpha helical. All right. Now let's look at some proteins with beta sheets. And so recall that the beta sheets can have residues above the sheet and below the sheet. And if it's above the sheet, say for example, they could all be hydrophobic. And then below the sheet, they can be all hydrophilic. So we can have amphipathic beta sheets where one face of that sheet is all hydrophobic and the other face is hydrophilic. So they can be amphipathic as well. And if we look at some proteins that basically have a lot of beta sheets, they're called beta domains or beta proteins. And if we look at these structures, and we basically find some common characteristics, is that the anti-parallel sheets often have one buried and one solvent exposed face. So the buried side will be hydrophobic, and the other side of that sheet is going to be hydrophilic. So mostly anti-parallel sheets are often amphipathic. Parallel sheets are pretty much always buried. So they are all hydrophobic. and they're buried. So they're alternating above and below the sheet are going to be hydrophobic all the way around. And so they'll be buried inside the core of the protein. Whereas the other sheets will be, half of it will be buried, half will be solvent exposed. That's the anti-parallel sheets. So here's an example of an all beta sheet protein, the immunoglobulin. Probably familiar with that and how our immune system works. And so you can see that there are 12 immunoglobulin domains here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. These are held together through disulfide bond linkages through cysteine residues, and that there's variable domains that vary in sequence that give the antibody specificity to bind different epitopes and help our immune system to be able to recognize non-self and foreign antigens that can be identified and then removed. And so it's through this variable recognition here. And so if we look at the structure of this immunoglobulin fold, you can see it's all beta sheet with interconnecting loops, and these are going to be anti-parallel. And so if we look at a side view of that, we can see that these two sheets, like two pieces of bread, will come together to form a sandwich, and we call this a beta sandwich. And in the meat of the sandwich here are these hydrophobic residues that are forming the hydrophobic interaction. And so that's how this immunoglobulin fold is stabilized, through the hydrophobic interactions of these amphipathic beta sheets to form this beta sandwich, which is a domain that's used repeatedly in the structure of the immunoglobulin fold. Proteins can also have a combination of both alpha and beta components. So triose phosphate isomerase, or TIM, is an example of this alpha-beta type domain or motif. And so you can see here how it's arranged with repeating units of beta, alpha, beta, alpha, beta, alpha, and so on. And that the sheets here, and if you look at the sheet in the center of this guy, is that they're all pointing in the same direction. So that makes it a parallel structure, and that these are all hydrophobic and are forming the hydrophobic core, whereas the helices are facing the inside and the outside, so they're amphipathic. Half of them will be hydrophobic, interacting with the sheet. So these strands are interacting with the other strands here to form this interior, and the hydrophobic residues on the other side of the sheet are interacting with the hydrophobic residues on these amphipathic helices to position the helical regions, so that the hydrophilic components of these helical regions are facing the solvent. All right. This is a common fold, and about 10% of the enzyme structures have this fold. So you'll see these repeating units of structure over and over again if you study protein structure and do research in that. So there's only certain conformations that are going to be promoted in terms of protein folding based on the psi-phi angles, the size of the side chains, the amino acid sequence. So I have a tendency to see certain folds over and over again, especially if I have a functional role. You can reuse those folds for in other proteins. So the domains can be used in multiple ways by other proteins. I wanted to mention the proteins that span lipid bilayers, or the membrane proteins. And we'll talk more about this when we get to signal transduction and electron transport and oxidative phosphorylation. There's other topics in this course. But as far as structure-wise, there's two types. There's the alpha helical bundle, and there's the beta barrel, which is just basically, you can imagine a sheet, like a piece of paper, and you roll it up to form a cylinder. That's basically what's happening here. And these proteins are kind of inside out, because on the inside are hydrophilic residues to allow things to span across the membrane. And the things that are hydrophobic are on the outside because they're interacting with the lipid tail components of the bilayer. And the same with the beta barrel. You have hydrophilic residues on the inside, so you allow things to span across like water, like beta barrels for porins, for aquaporins that you see to transport water across will be beta barrels. And the outside of this are hydrophobic residues that interact with the lipid tails. And so these proteins are kind of like inside out relative to the proteins that are water soluble. But the same principles apply. I to I plus 4 and hydrogen bonding for the alpha helical regions, parallel or anti-parallel beta sheets. It's all the same stuff. It's just that the side chains are reversed in terms of inside versus outside, which it makes sense because they're residing in a lipid environment, not in an aqueous environment. Control and modulation is all often through these loop regions, which combine different molecules to allow signals to be transduced from one side of the membrane into the other, or to help facilitate transport of molecules from one side of the membrane to the other. So know these two, the helical bundle and the beta barrel. We finally made it to quaternary structure, and this is basically where we're looking at the assembly of multiple protein chains into a functional unit. So you can have a protein that's folded, it's got a tertiary structure, and then you accumulate a couple of these different proteins to form higher order structures. And so we should be showing examples of that throughout the course. So that immunoglobulin that I showed before, those have different domains, but they're covalently linked through the peptide chain. They're not separate entities. These would be separate entities that can dissociate and reassociate together, but they will associate to form higher ordered structures. But you could break them apart if you provide certain conditions that would facilitate that. Whereas the immunoglobulins, they're not going to those immunoglobulin domains aren't going to dissociate. They're covalently linked to one another. So that's the difference here. We're looking at the assembly of multiple proteins. And you need to kind of know some of the nomenclature. So this is something you need to make sure you understand, that you can assemble more than one polypeptide chain. This would be called an oligomer. And the individual units are called monomers or subunits. And that's the individual chain. So if you have a monomer, you just have one polypeptide chain that folds into a protein, into a tertiary structure. That's a monomer. You can have two monomers come together, and that would be called a dimer, 3 of them, a trimer, 4, tetramer, and so on, pentamer, hexamer, etc. We would call all of these oligomers. So when I think of oligomers, I think of more than just a couple. I think of a lot more. So often you'll use oligomeric if it's more than 6. But if it's a defined 6 or 5, you'll call it a hexamer or a pentamer or a tetramer. And oligomer is usually kind of more than 6 often is the case. But technically it's only more than 1. So any of those, dimers, trimers, tetramers, pentamers, hexamers, can be considered as a classification of oligomers. And the different types of oligomers can be of 2 types in terms of their composition. One, they could be homo-oligomers, where all of the monomeric units are the same, or they could be hetero-oligomers, where it's composed of different monomers. So for a dimer, if it were a homodimer, it would have 2 identical monomers. If it were a heterodimer, it would be 2 different monomers. The monomers must be able to interact, so they can't be too different from each other in terms of structure, but they must be able to interact in such a way that would do that. So a lot of transcription factors that control gene regulation is a promoter binding a promoter region could be a homodimer or a heterodimer, but the fact that it can dimerize in 2 different ways between homodimerization and heterodimerization gives it a lot more conformational, a lot more combinatorial ability to activate different genes. So you can say I have 12 monomers here, and they can either form homo or heterodimers. You can have a lot more combinations available through the aspect of heterodimerization than you would with homodimerization. And that's often the case, is that families of transcription factors will be able to heterodimerize to promote more gene control through this heterodimerization. So let's just look at some examples of some of these quaternary structures. We're going to be talking quite a bit about hemoglobin, and that's shown here in this. It's not really a ribbon diagram, but you can see some alpha helical regions here on this protein, and there's no beta strands in hemoglobin, so it's all alpha helical, and you have different subunits. You have 2 alpha and 2 beta. These are monomeric units, and they're arranged in this way. Each can bind an oxygen molecule. We'll be going into that in detail. One of the proteins of HIV is a protease that helps to cleave the proteins that are made from the RNA that's encodes for the protease that helps to make mature virus, and it's an aspartic protease. We'll be talking about proteases in a different topic. And so in this case, it's a homodimer. In other words, you have a monomeric unit that comes together to form a homodimer, and the active site is actually components derived from each monomeric unit. So the monomeric unit by itself is not able to cleave any protein that's made, but the homodimer is. Here's another example as well with the capsid protein from bacteriophage, MS2. Again, that's like a virus of bacteria, bacteriophage. And it's also a homodimer. And in this case, a trimer, I was going to say, I see 3 units here. It's a trimer that allows it to be active as a capsid protein. So why do we have these higher order structures for? It just becomes more complicated. And we look at the reasons of what can be obtained through these quaternary structures that you can't do with tertiary structures. And that's what we're going to kind of finish up this topic with, is that the active site, or the ligand binding site, what it binds to, often occurs at the interface of the multimers. So the multimers by themselves can't do anything. I showed that example with the HIV protease, where you need the homodimer to form because it's contributing to the active site of the enzyme. For this binding of this zipper protein to DNA is basically phosphine-June. That's an important gene expression. And so you can see here there's these 2 alpha helical components. There's more to the proteins than just these 2 alpha helical regions. It's just not one giant alpha helix that's to form a coil, but this is part of the interaction, and there's a lot more stuff elsewhere. But this is what's allowing the side chains that recognize these parts of the DNA and the base pairs to allow specificity based on the promoter region, the DNA specification of the promoter to activate this gene. And so you need both of these to contribute. One's not going to be able to bind specifically enough, it'll just fall off. You need the other one to help recognize that sequence and to bind. So the interface is at the what is required for recognizing that DNA sequence. So you need both parts. One by itself is not enough. The second part has to do in terms of what the quaternary structure can provide is regulation. So binding of or an alteration in the structure of one of the monomeric units can have an effect on the other units of that complex. And so we call this an allosteric effect or cooperation in terms of regulation. And we'll be talking more about this when the topic of myoglobin and hemoglobin is presented. So I just want to introduce the concept here of an allosteric effector. So for our hemoglobin, where we have 2 alpha and 2 beta subunits, this 2,3-bisphosphoglycerate, a small molecule, will bind at the interface between these subunits, causing a conformational change that has an effect on hemoglobin to help stimulate the release of oxygen. And so if it were monomeric, this molecule has no effect on the release of oxygen. Whereas if it's a alpha 2, beta 2, a dimer of dimers, or a tetramer complex, this molecule 2,3-bisphosphoglycerate can bind at the interface that exists, causing subtle conformational changes that help to release oxygen. So we'll talk more about that in hemoglobin, but this allosteric control is important, and a lot of our enzymes that we look at in terms of metabolism are often not just monomers, but sometimes they're dimers, trimers, or even hexamers that will have these allosteric effectors that will help regulate enzyme activity. The other aspect is that this oligomerization again can cause huge molecular machines to be constructed, and that's shown here in the ribosome. The ribosome. The ribozyme is a catalytically active nucleic acid, and you'll learn about that in nucleic acid section. But this is the ribosome, which is used in translating mRNA into a protein, and it has 54 proteins and 3 RNA molecules as part of this huge complex. And so because it has so many proteins, it can be modulated in a lot of different ways here to basically recognize the mRNA in order to bring in the proper amino acids in order to catalyze the formation of the peptide bond and promote the growing polypeptide chain. And as the chain grows, it starts to fold and form the three-dimensional structure of the protein as it's being translated. So these molecular machines are only possible through these multiple interactions with monomeric subunits. So that wraps up the higher order structures of proteins. So the examples I provided are there for you to get a feel for the importance of some of these domains we discussed in the proteins and their higher order structures. So have a good feel for understanding that, but there's not, and use the, don't memorize all the details, but follow the learning objectives and hope that guide you in your studies, because we'll show many examples of this throughout the rest of the topics in the course. And so it's good to understand the terminology here and some of the aspects involved in the regulation of protein and their function.